the main reason we're here, which is to hear from Phil Starks. He's a professor of biology at Tufts University, and he's worked on a wide range of topics in behavioral ecology and also on a wide range of organisms. I think especially of work on invertebrates, insects, when I think of Phil and his work. He received his PhD from Cornell in 1999, and then went on to a Miller Fellowship at Berkeley, and that's where I overlapped with Phil for a few years when I was in the Berkeley area and UC Davis area, and got to know him a little bit there. And today he's going to be talking about a commentary piece that he published in Evolution Medicine and Public Health, EMPH. And for those of you who don't know, these commentaries are designed as a way to present a new hypothesis. And very importantly, what evidence is there for that hypothesis, and what directions are there for really testing that hypothesis and any alternative hypotheses related to some phenomenon. So it's a section of the journal we really like. And he's going to be talking about one of these commentaries today that he published. And so with that, I'll turn things over to Phil. It's great to have you here, Phil.
Hi, thanks. Thanks so much for inviting me. I'm very excited to give this presentation, and I'm super excited to see Peter Nonax out in the audience. He's a former co-author of mine. That's very exciting. So let me share my my screen, and we can start. So yes, this is my title today, HIV and male sexual behavior. Mostly going to take you through the hypothesis, but at the end, I'd also like to go through its history, as this is a project that really has lived in my to-do pile for many, many years. I am speaking from home, and this is vacation week. So there is a non-zero probability that either of these two little monsters will make it into the back of the room. Last I saw, they were dressed, so I'm assuming they'll be dressed if they appear. If they're not, please just avert your eyes. These are my two little beautiful children, William and Matthew. They almost look like twins, but they're 2.5 years apart. And thank you, Charlie, for that introduction. So one of the reasons that I'm a little bit more nervous than normal giving a talk like this is that I don't spend very much time studying humans or viruses or sexual behavior in humans or any of these things. I am a behavioral ecologist who really spends most of my time studying social insects, either these gorgeous beasts here or these ones. My lab has gone off in a lot of different directions. But most recently, we're focusing on group level adaptations within this global climate change environment and thinking about things like nutritional ecology and invasion biology. So this is what I would normally talk about. And probably the best thing about being a behavioral ecologist, like I am, is I get to teach animal behavior. And animal behavior is probably the greatest course that you can teach. I'm biased, obviously, and I don't think that my course is the best one you'd ever take, but it is a course that really can fundamentally change the way you look at nature. And that's cool. So I get a big kick out of that. I also benefit because humans are behaving animals. So students already have a feel for the material that you're covering. And you can teach them about a lot of things. And one of the things that I teach my students about from time to time are zombies, because everyone likes zombies. Zombies are exciting. Zombies are terrifying. But the idea of having a body taken over by something else is something that students can really wrap their mind around. They get a lot of interest in it. I don't want to say a big kick, a lot of interest in it. Right, so we spend a lot of time thinking about how, especially pathogens, how things get from one host to another, all the clever strategies. And so I talk to my students about this an awful lot. And I'm sure this audience is very familiar with these topics. So if I want to talk about Toxoplasma gondii, for example, the students find it fascinating that a mouse is the intermediate host, with the final host being cats. They find it interesting that once this little single cell pathogen infects the mouse, it changes the mouse behavior. The mouse becomes more excitable. It covers larger territories. It has an odd interest in the smell of cat urine. And you can imagine that these things are not particularly good for the mouse. It's really, really good for the pathogen. This is the way it ends up in the final host. And of course, if you follow the literature on this closely, you'll notice that a lot of humans are infected with this pathogen, and it may have some significant behavioral changes with us, both individually and maybe more globally from country to country. But this is a topic that my students really enjoy. And this is another fantastic topic. This is a fungus. How you just have to love this, right? And the ant becomes infected with the fungus. The ant climbs up a little tree, attaches itself to the bottom of a leaf. The fungus, it grows through the animal and eventually sporulates, being able to infect individuals underneath it. Now, this is very unusual behavior for the ant, right? But it's extremely good behavior for the fungus. So here we've got a single cell pathogen. Here we've got fungus that are infecting ants and changing their behavior. So the students enjoy this. This is this is just a virus, right? Impacting, in this case, a caterpillar. And caterpillar is going to climb to the top of a branch or tree. It's going to attach itself and then essentially dissolve so that the viral particles can be released below. So this is just a virus, right? Just some DNA that's impacting the behavior of the host. And we see this with encephalitis too, right? So there's some nice studies showing the impact of this virus on mouse behavior, how it increases the aggression, the territory size of the animal, how it increases the likelihood that it'll come in contact with other potential hosts. And we know the classic example here for viruses is rabies, right? So now I've taken you, so I don't need to give you a lot of details on this, but the rabies virus is extremely good at modifying host behavior and physiology to some extent to facilitate its spread into the next host, right? So now I've just, talk to you about a few very simple examples, right? In a caterpillar and some mice, here in mammals and in humans. And so it's clear to see, right, that, and this is a very simple hypothesis, like from an evolutionary perspective, pathogens that alter host behavior in ways that increase transmission have an advantage over pathogens that do not. That's just clear. So I asked the question, can a virus alter human sexual behavior? And this is a little bit of a scary question because, one, it's humans, and I don't think that humans particularly like the idea that we can be manipulated. And two, it's about something very personal to us, our sexual behavior. But this is the question that I wanted to ask. And so I'm going to ask that question, obviously, in HIV, and it's useful to think about what's happening with both your immune system and with the viral load, right? So where I really want to focus, right, is here in this acute phase, right? We'll call this acute and early phase, and here clinical latency, right? So this is when the body is actually keeping the viral load a little bit lower. But here in this initial phase, where you go through this large peak with your viral load, and this is a time when individuals may not know that they're infected, right? So this is the phase that we're focusing on. And so we have three general phases here, the acute and the early stage, the chronic stage, and then the advanced stage, AIDS. And HIV can be transmitted during all of these phases, right? But the acute and early stages where you have extremely high viral loads, right, and individuals may be unaware of their status, are likely to are likely to have the most profound impact. And indeed, in the acute stage of HIV infection, individuals are especially infectious. So we wanted to study this hypothesis, a hypothesis that HIV is manipulating host behavior during the early stages of infection. So how do you test a hypothesis like this? Right. So I think this is a big challenge. Like, for instance, I don't think you're going to get it past IACUC, right? So this is a tough thing to do ethically in any populations. But what you can do is you can look at viral loads in individuals, and you can look at self-report data on their sexual behavior. And there is some of that data out there. So here from Dukers in 2001, we've got on the y-axis the rate of unprotected sex with casual partners. And along the x, we've got the viral load. Okay, and if you look, you can see that as viral load increases, you get a non-linear increase, right, with the rate of unprotected sex with casual partners. So this suggests that there's some level of viral load that may influence risky sexual behavior, and maybe an increase in the number of partners. And this wasn't the only data from even from 2001. So here, this is Kalichman, and here we're looking at the number of insertive acts, right? Again, by the viral load. Now, what's different than this one in particular is this is the semen viral load, and the other was the blood viral load, but both show the same general type of behavior. Around log 5, you end up with a significant increase in insertive acts. So that tells me a little bit about how the virus may influence individuals who are already infected, right? And the viral load may have some impact on individuals who are already infected. But it really doesn't tell me the first part of my hypothesis, right? Does HIV manipulate human sexual behavior initially? And this is, of course, that chicken or the egg problem, right? The answer can always be, listen, individuals who have naturally high levels of sexual behavior are more likely to be infected. And thus it seems that they've got, unless it's the chicken or egg, we don't know that it's the virus or if it is the individual. But we can get around that just a little bit. And this is a very interesting paper. This came out, I think, in 2020 by Joseph Davey. And what we're looking at here is individuals who are in the acute infection, and we're comparing it with individuals in the non-acute infection stage. So just to remind you, right here, we've got one of these initial, this is acute. This is a short period of time, just a few weeks. And then here we've got the non-acute, which can be extended for years. So we're looking at two very different time frames. And here, what the researchers were looking at is the number of sex partners and by time, like whether it was in the past 30 days, was in the previous 3 months, and again, breaking it down by these different individuals of acutely, people in the acute infection stage or the non-acute infectious stage. And I want you to pay attention to these data in this comparison. So in all cases, right, the individuals in the acute infection stage had more sex partners in the previous 30 days, had more sex partners in the previous 3 months. They were more open to receptive and insertive acts over the previous 3 months than individuals that were in the non-acute phase, so later on in the infection. So this pretty much tells you the same story that the other figures I put up tell you, right? So you're likely to have higher viral load in the acute infection stage, higher viral load there than in the non-acute stage. So this is, again, like with increased viral load, you're likely to have increased sexual behavior. But this was the part here that really jumped out, right? So for you to be in the acute infection stage, you likely converted within the previous month. So within that 30-day period, right? So likely the 2 months previous, right? This is unfortunately this month plus the 2 previous months, right? Tell us a little bit of a story of the mean number of sexual acts an individual is likely to have before becoming infected and then after becoming infected. And in this case, when you do the simple math, you're almost doubling the amount of sex you're having in the month that you convert. So from a much lower rate prior to conversion to a higher rate post-conversion. And so again, very briefly, I feel that these data do suggest that a virus can alter the human sexual behavior, that viral load can alter the types of behavior, how risky that behavior is, and that there is some suggested evidence that the infection itself increases sexual behavior early in the infection. Now, how is a different question. And how is a big question here. So I made a hypothesis on how HIV infection might be impacting sexual behavior. And I did that back in 2000. And so you can tell because the figures are much rougher here than the other ones that I put up. So this is from just an abstract that I published in AIDS 2000. And we hypothesized, again, the same hypothesis that HIV infection might be influencing human sexual behavior. And here we didn't have the data on viral load and sexual behavior, but we did have data on testosterone, which can be a correlate of sexual behavior. And in with these, we had 7 studies that showed the data, and there was a relationship, a positive relationship, where individuals with infection had a higher total testosterone than those without the infection. Now, this is a really big problem with respect to the chicken or egg because we have no idea. So this is just a correlate. This is a potential mechanism for how sexual behavior is influenced, but it's just speculative. I'm telling you that really more to go back into the history of this idea. And I kind of want to know if it resonates with any of you doing research. So this is the general idea we have anyway, that HIV can manipulate the host behavior and it benefits by the host's increased sexual behavior in terms of getting into future hosts. The proximate mechanism could be testosterone, it could be a number of things. I'm uncertain. But this was a hypothesis that was born in 1995, I think in the spring of my first year in graduate school, or it could have been the fall of my second year of graduate school. And it was born here in Seeley G. Mudd Hall, where Paul Sherman took, I see Randy's on the Zoom, so I'm excited about that, decided that he would teach his first course on Darwinian medicine after that fantastic book came out. And so I developed the hypothesis here. It was a broader hypothesis at the time. I was really looking at the rate of, or the duration of being asymptomatic and the likelihood of a single per contact infection with a partner. But those data were hard to come by. But I didn't drop this hypothesis at that stage, but I did end up graduating, thank God. And then I ended up in, like what Charlie was saying, out here in Berkeley. And this is where the paper grew a little bit. This is where we submitted the abstract. And that is where things got weird for me. Right? I had recently published another paper on human behavior, and that one received an interesting response. There was some rather ridiculous press coverage, and there was a paper written, kind of an attack piece written by my former colleagues at Cornell that thankfully didn't get published. And people that I really, really liked congratulated me on committing career suicide. So this was truly shocking. So this is how I felt, very much like this. And you know, but the idea didn't disappear. So it kind of hung with me for a bunch of years. And I went through a number of really outstanding students. So two of them, David and Maxfield, they, you know, I would share this idea with them and they would come back. But I think that I was probably never going to work on this again until I got tenure, right? Because I kind of believed in that career suicide aspect. And I may not have come to it again. I may not have come back to this project again if I didn't feel like there might be some value in this hypothesis beyond just, you know, identifying something that may be manipulating our host behavior. I'm hoping that someone will pick up this research. I'm hoping that someone will understand the mechanism if it exists. I'm hoping someone will disprove it if I'm completely wrong. But if it is correct, then it really does argue in favor of increased testing. And it may even argue that individuals who are positive may want to may want to get additional tests if they are feeling particularly adventurous, let's say. So I think that there might be some value in that hypothesis. And there, I told you, Charlie, it'd be short, right? So I just want to thank my co-authors, Maxfield and David and Wayne Getz, who's fearless. My students, Isaac and Jesse, who put up with this stuff right now. Those are my current students, many students in the past. And I want to thank all of I stole Sherman's class on Darwinian medicine. I've been teaching it every fall since 2003. So I want to thank all those students who often kicked in. And I want to thank my good luck. I want to thank that Charlie wanted to get that commentary piece reviewed by some experts. I'm very happy about that. I want to thank Meredith, who didn't give up on me, because I can be a nightmare to get in touch with. And I want to thank this audience for your time and attention today. So thank you. I think I'll stop the share and see some faces.
Fantastic, Phil. Thank you very much.
Was that the right length?
That was perfect. That was perfect. And there are a ton of questions in the chat box. I think it's going to be challenging for us to sort through these. I would like to ask people to raise their hands if they'd like to ask a question. And because I'm going to tune out early to teach, I thought I'd just ask one question really fast to start things off. If I understand correctly, the acute phase is associated with pretty severe flu-like symptoms, the acute phase of HIV. I mean, so much so that people often report to, you know, their primary care physician. And that doesn't seem to be a time when people would be, you know, interested in seeking out partners and going out and socializing and doing all these kind of things that would be associated with that. I'm just wondering, that seems to not quite square with, maybe my understanding's wrong, but how does that, you know? No,
your understanding's good, Charlie. It's and I've had the flu, and I've never felt less attractive than when I had the flu. There was no way that I was looking for anything outside of some chicken soup or something, right? But that stage, it doesn't extend for the full 3-4 weeks of the initial infection. Right. So yes, there are some initial flu-like symptoms, but they tend not to linger throughout the full acute phase. But if anyone has better information, please jump in. I would love to hear it. I am, you know, again, I'm a bee guy for the most part, a wasp guy, right? So if you have better information, I'd love to hear it. I think I saw Pete Nonax's hand up. Pete?
Yeah, so Peter Nonax, if you're there and have a question, feel free to unmute and go ahead and ask.
I'm unmuting myself and here, and it's remarkable how mirror image we are, in a sense that, you know, I'm very glad that your qualities kept you from being murdered in your scientific career. I think that's a benefit for all of us. But just like you, I'm a behavioral ecologist, and my hubris is large, and I also sort of go to other fields and try to explain those fields to experts in those fields and get the same kind of like, who the hell are you reaction. And so we actually also, as you know, one of the things that I've worked on in social insects is the advantages of genetic diversity rather than kin selection and genetic similarity, the advantage of genetic diversity, and now how that helps groups be more productive and therefore can be selected for. And so myself and a student, like in 2008, sort of, well, where can we actually test this? And we actually did models in HIV. And the same kind of thing is like, you know, why does it take a decade for the onset of AIDS? Why is this thing so, you know, sort of chronic and low level and suddenly explodes? And our feelings were that what you have here is an RNA virus that basically evolves within its host. It becomes a quasi-species, swarms, and it oftentimes co-infects cells. So in other words, rather than sort of viewing all of these clones as being competitive, you could actually view them as being, in a sense, cooperative. So when they co-infect, they're actually more like polyploid chromosomes interacting with each other rather than competitors. And so we modeled this, we came up with, I think, a lot of results from the model that fit with sort of the progression of HIV to AIDS. And it took us over 4 years to publish that paper because we kept getting rejected from one journal to another journal to another. Sometimes I would say for good reasons. You know, we got plenty of good comments back, but we also got the sort of comment of, you guys need to put a famous virologist on this paper. You know, it's like, okay, who are you to tell virologists how HIV works? Until you actually get a credible name on this paper, you're not going to publish it. So yeah, we finally published it in Evolutionary Medicine, your sort of competing evolutionary medicine journal. And we actually have a second paper right now that's out in submission, and we'll see how it goes, from a talented graduate student that I had, to expand this and actually look at over 100 different studies in viruses, mostly RNA viruses, and cancers, to show again that the genetic heterogeneity actually correlates oftentimes very strongly with the virulence or the nastiness of either the tumor or the viral infection. And we'll see how that goes. You know, we don't know. So just to sort of let you know that, been there, done that. Yeah, I know what you're going through. It's tough when you sort of come from a different field. Hang in there and do it. And the other quick comment I'll make is that I also have to leave, like Charlie, at 40 minutes after to go teach a class. So I'm not leaving because you've offended me or anything. I just have to go. So now I'll let other people make comments and questions.
It was my goal to offend you at one point, Pete, so I'm unfortunately I failed. But yeah, no, so I'll tell you that I've been pleased. Sorry, Charlie. I was pleased that I haven't received a lot of feedback on this paper at this stage. So I think, you know, the pandemic might have helped or whatever, but it's a little bit slid under the radar so far. It doesn't really work for my long-term goal. I'd like to follow up with what you were just saying, Pete, because one of my initial ideas in these studies was that the degree that an individual would remain asymptomatic, right, is influenced by how easy it is on a per contact basis to get into another host, right? So what I originally wanted to do was to look at a lot of different sexually transmitted infections and see the per contact transmission rate. And with HIV, it's exceptionally low for like a single contact, which made sense with the original idea that it would have such a long asymptomatic period. But that's an idea that I've dropped. Anyone else in this room can pick that one up if no one's done it. Diana, I think you had your hand up. Is that right?
Yeah. Next, we'll go to Diana Fleischman.
Hi, I'm Diana. And yeah, I've been working on this idea for a while with a couple of co-authors, the idea that sexually transmitted infections motivate sexual behavior. And looking at syphilis seems to induce a kind of mania. There's a very cool passage from Oliver Sacks about 2 different patients he had with syphilitic mania, and including a 88 or 90-year-old woman who felt this increase in sexual behavior. And it turned out that she had a resurgence of neurosyphilis because she was never treated completely. So I wrote a blog about it, I could put it in the chat. But I was wondering if there's any evidence that HIV actually has neurological effects. I tried to find it in your paper, but maybe you didn't have enough space to talk about that. So yeah, is there evidence that there's neurological effects or neuroendocrine, other neuroendocrine effects? Yeah.
I believe absolutely yes. It's not something that I dove into in a lot of detail because I didn't want to get into the how it impacted. I didn't want to get into the proximate mechanisms of the influence on behavior. I thought that that would be best left for somebody who's trained in that area. I was more looking at the general the sexual behavior data. But yes, you will find it there, Diana. So if you guys want to raise hands, I don't know if you go in restriction under reactions now down below is where you can find your hands. James.
Hi, yeah, so I have a question just because one is about a clarification. It's kind of a two-parter. And the second is more about just like categories. So the first one is about like, was the data evidence that you presented, was it only on kind of sexual minority males, or was it only on, or were there cisgender heterosexual males also kind of in the data?
So I think most of the data in the paper are on men who have sex with other men. Okay. Right. So I think a lot of the data were collected at a center that is more broad than that. But I think that's the data that was originally published on. And so there is another study mentioned in the actual paper that I wrote that I didn't go into today because the figures are really hard to understand, but that did include individuals who had sex with women, males who had sex with women. But for this entire project, I'm focusing on male sexual behavior.
Okay, so I guess my question then is how, because MSM is a behavioral construct, right? It's about who you have sex with rather than, and so that also lumps in trans women and people who are gender diverse. And so how do, like, I think that would be a big confounder, because if you're kind of suggesting, because of the relationship between testosterone and sexual behavior. So how, sure. Like that, obviously that's an important thing to control for.
Sure.
And so how would like a sex and gender based analysis kind of change the implication, or the potentially, like, what are the implications of that? Like also looking at gender diversity within your sample?
Well, that's a great question, James, really. And the problem is that they're not my samples, right? So these are papers that were published and data that I collected or that I took from those papers. I think I don't think that I'd want to hazard a guess on the different mechanisms on what might be happening, and I certainly don't want to get into. I don't want to get into it, and I know that we do this because it's, we're just looking at men with men, right? I don't want to get into too much of an identity aspect to the research, right? And that's mostly because I think that the research is lagging on those issues, and including that type of confounding variable in the data. Does that make sense, James?
Sure. Well, the thing is, like, I'm wondering how, like, being on like testosterone blockers and then receiving like estrogen would have influenced the kind of behavioral patterns that you would see from this data, right? So like that's the kind of that's what I meant by confounding. Not so much it's identity again related, but it's more so it's the implications that those identities have for the biology of the individuals.
Well, that's fantastic. That might be a really excellent way of disproving the hypothesis that testosterone is a driver, right? So, if individuals are on testosterone blockers and you still have elevated sexual behavior, then you might be thinking that Starks's little hypothesized mechanism is incorrect. That works for me. Science is cool, right? We destroy things by moving ahead. Steve.
Hi.
Hi.
Thanks. Thanks. I think it's a really compelling hypothesis that there's sort of good theoretical reasons to think is likely. I'm coming at this from being a sort of HIV epidemiologist who also works a little on sort of how sexual networks of MSM influence the evolution of the virus. And so I'm trying to, when I sort of looked at the studies you were doing, as a good scientist, my thought is, okay, are there more parsimonious explanations for these patterns so that you don't have to, before you get to kind of the evolutionary hypothesis? I suppose it depends on what you think is more parsimonious. But so in the third, and I was sort of looking at the first two studies in a group, and then the third study you presented from 2020, who's I don't remember the author's name, but in
Bishop
the
Davies.
Okay, in the third study, first of all, so in our group, we usually, I'm curious how they defined acute infection, like the duration of that, because we often talk about it in our group as being 3 months, sort of, so that there's kind of this biphasic decline in viral load. So even though there's the sort of, and there's a lot of variation among people. So even though maybe the first month is when it's super high, that kind of continues to be fairly elevated for up to 3 months. So sort of with that in mind, then the sort of 30-day, 3-month distinction becomes a little less clear. And then you have the idea that's, that's been batted around in the literature for a while, that men have a lot of heterogeneity over time in how many partners they have. They go on, when you're young or, at certain ages, or you're on vacation or you're not in a main relationship, you might have lots of partners. Having lots of partners predicts that you'll get HIV positive, and then if you happen to be in that phase of high activity, then you'll continue to have lots of partners up until you're diagnosed. So that's sort of always the trick where you have to control for. And what I liked about the studies is you were trying to control for that in different ways, and by looking at the different studies, but I just wonder, in that third study, the duration, whether a different duration definition of acute infections, kind of how that fits in there. And then a separate question on the first two, but
So they did it by viral load.
Okay.
I think so. So it was by viral load. And what's interesting in that study is that individuals, filled out the data and were tested, right? And they've discovered then that they were infected.
That's why it makes it so compelling, that you have the control. They didn't know yet, so they don't do it. So that part is really great.
Yeah, but the other parts, I understand that you would, the whole chicken and egg thing is a really, really big issue here. But I wouldn't want to say that mechanisms are better or worse explanations than evolutionary ones. It's just a different approach, right? And so I view that, Sherman was on my PhD committee, so I'm very much a levels of analysis guy, even to today. So I like thinking about these things as being mutually illuminating instead of mutually exclusive.
I didn't use the word better or worse. I said parsimonious.
Well, then I made a different argument.
Yeah. For the first two studies, could you tell whether those partner counts that you were looking at, that you showed, because it went by quickly, and it probably was on the slide, but could you tell if were those partner counts from before they were infected or after they were infected, or you can't tell? It's sort of all mixed. The timeline is mixed.
A lot of the timelines are mixed there, right? So it's harder to tell. What is interesting is some of them are with casual partners, right? Which is, so that's just another little tidbit of information there on that. Thomas.
Yes. Thank you. Interesting talk. I just wanted to share some anecdotal observations. During my many years of living in Uganda, when AIDS first became apparent in the early 80s, and when it really reached its peak, one heard different kinds of stories, and it relates mainly to the way that the people who had AIDS felt and behaved. In a sense, it talks about these anecdotes really address the issue of how these people are thinking when they get involved in this. And there were many accounts, and I don't know how many of them were ever proven, that once a person, and these were mainly, of course, heterosexual interactions, not much, as far as anyone knows, was due to homosexual interactions. So mainly heterosexuals. And so when a person discovered that they had AIDS, they would often become very angry. And their intent was to pass it on to as many people as they could as form of revenge to life, or whatever you want to call it. That was one kind of stories you kept hearing, and people were even convicted and put in prison with it. The other one was a guy I knew, a young man, heterosexual all the way. And I warned him about this. I said, you got to start using a condom. And he says, oh, this is so exciting. This is more exciting than sleeping with another man's wife. So the idea was it was a challenge. What the hell can I get away with? And I wonder if you have looked into sort of the stories people tell you as to why they do these things. So, you get an increase in sex. Is it, the assumption you're making is it's something that's sort of intrinsic. The virus maybe acts somehow neurologically, and maybe this is the way it's manifest. I don't know, but just a question to think about.
Yeah, I think the argument I'm making, or I'm suggesting, I don't want to make an argument. I'm suggesting that the virus would facilitate its likelihood of getting into another individual if it could upregulate, I guess, the sexual behavior of a host. But just to be honest, I don't like people very much, and I like you fine and everything like that, but I don't want to study human sexual behavior and what stories we tell ourselves or whatever about our own proclivities. There are people that do that very well. That's just not something that I care to invest in. I like my bugs. I like bugs. They're a little bit easier to understand. They lie less, right? So they're the ones that I kind of prefer working with.
But I guess I just thought it might be, if you might know of any studies that we're looking at the reported motivations behind people who got more involved in this, because I guess the homosexual guys you're talking about knew they were having more sex with more partners, and it was unprotected. And I guess the assumption was they thought that they were not infected. Is that correct? I mean, what do we know about infected individuals who know they're infected and go ahead with more partners more frequently?
Yeah, I think that's interesting. I don't, the hypothesis that I set out does not suggest that people are cognitively aware, right, of their infection, right? So it could just very well be that they feel good, right? People who feel healthy, they feel fine, they feel good, tend to have more sex than people who don't feel so good. That's why Charlie asked that first question, right? So about the flu-like symptoms. So I think my working hypothesis would be that people were unaware of their status and were not purposefully infecting anybody. That was just a consequence. Yeah. But the criminal behavior is just a terrible thing to think about, and I feel bad for the people like that.
I could not believe someone could do that, but yeah, people were talking about it everywhere that this was going on. Revenge.
Yeah, so I think there's good evidence that when people are aware of their infection, they then reduce their sexual behaviors or have safer sexual behaviors from other studies. So we'll move on next to a question that Adam Hunt just posted in the chat. Would retroviral drugs reduce sexual behavior in this hypothesis? Because that seems like something that you could test pretty easily.
That's really interesting. I would think yes. So in one of the studies that I put up, it showed whether or not people were on ART, and some people that are still end up having high viral loads, and they still show the sexual behavior, right? Increase in sexual behavior. That's the I think it's the Kalichman paper, 2001.
Great, thank you. So we also had another great question in the chat, and we probably have time for a couple more questions after this. If you do have final questions, feel free to raise your hand to get in the queue. But Jeffrey Miller had posted in the chat, apart from outright behavioral manipulation, there are anecdotal reports that HSV oral herpes often flares up as cold sores before first dates or when people first interact with new mates. So does anyone know if there's any evidence that viruses can track mating opportunities somehow and increase their transmissibility without necessarily impacting the host behavior?
How rude! That I honestly, that is outside. Jeffrey, I would love if you would talk a little bit about that, because that's fascinating to me.
Yeah, so I thought a lot about this because I have a PhD student, Ruth Serafin, who just did a dissertation on all of this, and she tried to do an empirical analysis of national data set, ad health. We didn't really find much, to be honest, but I think we did have a few interesting ideas, like just the fact that, if you're a virus like HIV or HSV, if you can at all increase your transmissibility, like migrate down neurons so that you're expressed in body surfaces and might come into contact with potential mates, then really it's only worth doing that when you can actually infect a new mate. You don't necessarily want to bother doing that with a long-term existing mate. So even the symptoms of these STIs should, optimally track what are your transmission opportunities. And it could even do that in a relatively sort of preemptive way, like even before, actual sexual behavior works, if you're gearing up your mating effort hormonally in various ways, that might influence at least the transmissibility of the virus, if not the virus kind of manipulating you to. We had other, hypotheses like, well, obviously, HSV should turn people bisexual, lower their mating standards, make them more promiscuous, make them more polyamorous, blah, blah, blah. So there's a lot of more speculative stuff, but at least in terms of the virus sort of putting itself out there on the body surface, does that make sense?
Yeah. I can't answer that with respect to HIV or any other pathogens. I think it's a fascinating hypothesis. I mean, illnesses are good at getting into new hosts, or they would go locally extinct, right? So.
Great, thank you. So another question I had seen in the chat, I think from Grzynia Jasenska, was asking about what we know about other sexual infections, whether there's been any documented host manipulation by other STIs. And I know Diana mentioned some effects of syphilis, but do we know anything about other either human STIs or STIs in other animals?
I personally am unfamiliar. I remember originally thinking maybe we could study this, in particular, study it in some primates. But even then, I think you still run into the same ethical problems that we're dealing with, trying to study it really well in humans. How about the group? Yeah, this is a very smart group of people. Are you aware of any other impacts of other STIs on human behavior?
And you can feel free to just come off mute if you have something to add for this point. All right, so I'm not seeing anything, so maybe that's uncharted territory. So one final question I had, I was wondering, I know it's fairly limited just by lack of data that is available, but what do we know about whether there's any effects of HIV infection for women's sexual behavior? Is there anything known about that?
Yeah, I think very, very little. And I mean, there's an obvious bias in in the studies. I was fascinated by that question early on, especially in 2000, and I looked for the same type of correlates of sexual behavior in females, and I didn't find any. I was also looking at thinking about the transmission probabilities, because there are two things that could happen for the virus with a woman, right? You could give the virus to another partner, another sexual partner, or you could give the virus to your offspring, right? So the virus has the opportunity for different transmission styles, I guess. And I could not find any information on that. But that was again back in the 2000s, early 2000s.
Okay, great, thank you. Well, I'm not seeing any other raised hands, and we are getting close to the top of the hour. So thank you so much for this talk. This was excellent and really great conversation. So thank you to Phil for presenting, and thanks to all of you for attending.
Meredith, can you save the chat?
Yes, so the chat.
I would love to read it.
Yes, the chat is saved and will be sent out with a recording of this session to everyone that registered. Really quickly, before everyone heads out, I did want to share our upcoming events. So this is our Club EvMed website, and we have the event that we're at right now. But next week we have Robert Woods giving a presentation on, does evolutionary biology have anything to say about how we should use antibiotics? And that's going to have a pretty clinical focus and also have additional clinical commentary from John LaPuma and be moderated by Andrew Reed. So we're really looking forward to that. So I will send the recording of this to everyone soon. And again, thank you all for attending and hope to see you next week.